Concept 4.2. Carbon atoms can form diverse molecules by bonding to 4 other atoms. The key to an atom's chemical characteristics is its electron configuration. This configuration determines the kinds and number of bonds an atom will form with other atoms. The formation of bonds with carbon. Carbon has 6 electrons with 2 in the first electron shell and 4 in the second shell. Having 4 valence electrons in a shell that holds 8, carbon would have to donate or accept 4 electrons to complete its valence shell and become an ion. Instead, a carbon atom usually completes its valence shell by sharing its 4 electrons with other atoms in covalent bonds, so that 8 electrons are present. These bonds may include single and double covalent bonds. Each carbon atom thus acts as an intersection point from which a molecule can branch off in as many as 4 directions. This tetravalence is one facet of carbon's versatility that makes large, complex molecules possible. When a carbon atom forms 4 single covalent bonds, the arrangement of its 4 hybrid orbitals cause the bonds to angle toward the corners of an imaginary tetrahedron. The bond angles in methane are 109.5 degrees, and they are roughly the same in any group of atoms where carbon has 4 single bonds. For example, ethane is shaped like 2 overlapping tetrahedrons. In a molecule with more carbons, every grouping of a carbon bonded to 4 other atoms has a tetrahedral shape. But when 2 carbon atoms are joined by a double bond, all bonds around those carbons are in the same plane. For example, ethane is a flat molecule, its atoms all lie in the same plane. We find it convenient to write all the structural formulas as though the molecules represented were flat. But keep in mind that molecules are 3-dimensional and that the shape of a molecule often determines its function. The electron configuration of carbon gives its covalent compatibility with many different elements. The valences of carbon and its most frequent partners, oxygen, hydrogen, and nitrogen. These are the 4 major atomic compounds of organic molecules. These valences are the basis for the rules of covalent bonding in organic chemistry, the building code for the architecture of organic molecules. Let's consider how the rules of covalent bonding apply to carbon atoms with partners other than hydrogen. We'll look at 2 examples, the simple molecules carbon dioxide and urea. In the carbon dioxide molecule, a single carbon atom is joined to 2 atoms of oxygen by double covalent bonds. Each line in the structural formula represents a pair of shared electrons. The 2 double bonds formed by the carbon atom are the equivalent of 4 single covalent bonds. The arrangement completes the valence shells of all atoms in the molecule. Because CO2, or carbon dioxide, is a very simple molecule and lacks hydrogen, it is often considered inorganic, even though it contains carbon. Whether we call CO2 organic or inorganic, however, it is clearly important to the living world as a source of carbon for all organic molecules and organisms. Urea is the organic compound found in urine that Wohler synthesized in the early 1800s. The structural formula for urea is shown at the right. Again, each atom has the required number of covalent bonds. In this case, 1 carbon atom is involved in both single and double bonds. Urea and carbon dioxide are molecules with 1 carbon atom. But a carbon atom can also use 1 or more valence electrons to form covalent bonds to other carbon atoms, making the atoms into chains of seemingly infinite variety. Molecular diversity arising from carbon skeleton variation. Carbon chains form the skeletons of most organic molecules. The skeletons vary in length and may be straight, branched, or arranged in closed rings. Some carbon skeletons have double bonds, which vary in number and location. Such variation in carbon skeletons is one important source of the molecular complexity and diversity that characterize living matter. In addition, atoms of other elements can be bonded to the skeleton at available sites. Hydrocarbons. Hydrocarbons are organic molecules consisting of only 1 carbon and hydrogen. Atoms of hydrogen are attached to the carbon skeleton wherever electrons are available for covalent bonding. Hydrocarbons are the main compounds of petroleum, which is called a fossil fuel because it consists of the partially decomposed remains of organisms that lived millions of years ago. Although hydrocarbons are not prevalent in living organisms, many of a cell's organic molecules have regions consisting of only carbon and hydrogen. For example, the molecules known as fats have long hydrocarbon tails attached to a non-hydrocarbon component. Neither petroleum nor fat dissolves in water. Both are hydrophobic compounds because the great majority of their bonds are relatively nonpolar carbon to hydrogen linkages. Other characteristics of hydrocarbons is that they can undergo reactions that release a relatively large amount of energy. The gasoline that fuels a car consists of hydrocarbons, and the hydrocarbon tails of fat molecules serve as stored fuel for animal bodies. Isomers. Variation in the architecture of organic molecules can be seen in isomers, compounds that have the same number of atoms of the same elements but different structures and hence different properties. Compare, for example, the two 5-carbon compounds. Both have the molecular formula C5H12, but they differ in the covalent arrangement of their carbon skeletons. The skeleton is straight in one compound but branched in the other. We will examine 3 types of isomers: structural isomers, geometric isomers, and enantiomers. Structural isomers differ in the covalent arrangements of their atoms. The number of possible isomers increases tremendously as carbon skeletons increase in size. There are only 3 forms of C5H12, but there are 18 variations of CH18 and 366,319 possible structural isomers of C20H42. Structural isomers may also differ in the location of double bonds. Geometric isomers have the same covalent partnerships, but they differ in their spatial arrangements. The differences arise from the inflexibility of double bonds. Single bonds allow the atoms they join to rotate freely about the bond axis without changing the compound. In contrast, double bonds do not permit such rotation, resulting in the possibility of geometric isomers. If a double bond joins 2 carbon atoms and each C also has 2 different atoms or groups of atoms attached to it, then 2 distinct geometric isomers are possible. Consider a simple molecule with 2 double-bonded carbons, each which has an H and X attached to it. The arrangement with both X's on the same side of the double bond is called a cis isomer, and the arrangement with the X's on opposite sides is called a trans isomer. The subtle difference in shape between geometric isomers can dramatically affect the biological activities of organic molecules. For example, the biochemistry of vision involves a light-induced change of rhodopsin, a chemical compound in the eye, from the cis isomer to the trans isomer. Enantiomers are isomers that are mirror images of each other. In the ball and stick model shown in figure 4.7c, the middle carbon is called an asymmetric carbon because it is attached to 4 different atoms or groups of atoms. The 4 groups can be arranged in space around the asymmetric carbon in 2 different ways that are mirror images. Enantiomers are, in a way, left-handed and right-handed versions of the molecule. Just as a right hand won't fit into a left-handed glove, the working molecules in a cell can distinguish the 2 versions by shape. Usually one isomer is biologically active and the other is inactive. The concept of enantiomers is important in the pharmaceutical industry because the 2 enantiomers of a drug may not be equally effective. In some cases, one of the isomers may even produce harmful effects. This was the case with thalidomide, a drug prescribed for thousands of pregnant women in the late 1950s and early 1960s. The drug was a mixture of 2 enantiomers. One enantiomer reduced morning sickness, the desired effect, but the other caused severe birth defects. Unfortunately, even if the good thalidomide enantiomer is used in the purified form, some of it soon converts to the bad enantiomer in the patient's body. The differing effects of enantiomers in the body demonstrate that organisms are sensitive to even the most subtle variations in molecular architecture. Once again, we see that molecules have emergent properties that depend on the specific arrangement of their atoms. Now we'll move on to concept check 4.2. So we're going to skip the first 2 questions and go on with the third question. And the third question is, how are gasoline and fat chemically similar? Referring back to what we've learned so far, we can tell that both gasoline and fat consist largely of hydrocarbon chains. And so we'll move on to the next question, which is a what-if question. And the question is, can propane C3H8 form isomers? Well, the answer to that is no, because there is not enough diversity in the atoms. It can't form structural isomers because there is only one way for 3 carbons to attach to each other in a line. And so there are also no double bonds, so geometric isomers are not possible, and each carbon has at least 2 hydrogens attached to it. So the molecule is symmetrical and it cannot have enantiomeric isomers.